My name is Penny Ward. I happen to be chair of the Education and Standards Committee at the faculty, and it's my delight to be joined today by a very distinguished panel of experts in the field of clinical development and research in a variety of different settings. Dr. Susan Tansey, Mr. Wilfred Gurupira, Dr. Carol Shukovitz, and Dr. Gina Swartz. Susan, would you care to introduce yourself to the group?
Yes, hello. My name's Sue Tansey. I'm a consultant pharmaceutical physician working as an independent consultant currently. I have been in the pharmaceutical industry for over 20 years, working majority of that time in clinical trials in various therapeutic areas. Prior to that, I trained as a paediatrician in the NHS. And my particular interests are clinical trials, planning and strategy, and I'm also interested in ethics, and recently was appointed as a member of the Nuffield Council on Bioethics. So really looking forward to the discussion this afternoon. Thank you.
Gina, please.
Good afternoon, everyone. My name is Dr. Gina Swartz. I am a clinical neurologist and a neuroscientist with a PhD in molecular neurogenetics. I trained in internal medicine and neurology in South Africa and then came to the UK where I did my PhD at the University of Cambridge. I have also worked in industry for about 20 years now. I have always worked in the area of neuroscience, neurology and psychiatry, that being my area of expertise, either in global clinical development or in earlier development encompassing translational medicine or clinical pharmacology, but always in the context of developing new therapies in the space of neurodegeneration, neuroinflammation, stroke, etc. I also have retained academic and teaching affiliations, and I'm an honorary consultant at Imperial College London, where I have carried out cognitive disorder related clinics over the years. I also worked for the Foundation for Treatment of People with Victims of Torture, and I am also affiliated as a visiting lecturer at King's College London, where I deliver lectures on the pharmacology of neuroscience and clinical development in that context. So it's a pleasure to be with you today. Thank you.
Thank you, Gina. Wilfred, please introduce yourself to the attendees.
Good evening, everyone. My name is Wilfred Gurupira. I am a research pharmacist based in Harare, Zimbabwe. I work in HIV, AIDS, prevention and treatment clinical trials primarily. I've been working there in this space for the last 15 years. And I'm also currently studying towards a PhD in public health. My main area of focus is around complexity. And lately I've also been working with the African Alliance on the COVID research and advocacy project, really looking at community involvement and engagement in trials around COVID-19. Thank you.
Thank you, Wilfred. And Carol, would you introduce yourself to the attendees?
Sure. Thank you, Penny. Good afternoon, good evening, everyone. First of all, I'm very sorry, but I have some technical issues with my camera, so I cannot share. A few words about me. I'm in the clinical research industry for the last 12 years. I must admit that I worked in the past within the industry only, which means the pharmaceutical companies and CRO companies. And my experience is aligned with clinical part of the research only, with a wide range of indications. And currently, I'm a global project manager at Janssen, which is a company of Johnson & Johnson. And additionally, I'm a board member of the biggest association in Poland, which is Association for Good Clinical Practice. And I have a training section there, so I'm also a leader of many educational initiatives within the clinical trials industry.
Thank you, Carol. Well, welcome to our panel and also to all the attendees, and particular welcome to those of you who are not Faculty of Pharmaceutical Medicine members. The format of today's meeting is a discussion, a question and answer session, if you like, and a discussion between our panel members on the topic of clinical development of therapeutics in a pandemic preparedness setting. And I have a number of questions to pose to the panel, but for those of you who are listening to the discussion and wish to join in and pose your own question, you can see in the GoToMeeting box that there is a chat function, and please enter your message there, your question there, and we will try to make sure that those are addressed by our panel as the meeting progresses. Our session, as we heard earlier, is being recorded, and you can also chat if you can see other people that you want to have a short email exchange with in the attendee list. At the moment, we have 115 people online. So I'm going to start this topic on the topic of the repurposing of agents for the treatment of this global pandemic of COVID-19. And Gina, I particularly would like to start with you, given your background. When considering repurposing an agent for use with a new disease, what key pieces of information do you think should be generated prior to clinical investigation?
Well, I think it's very important to consider the approach that we have adopted. I omitted to say that I currently work as the therapeutic area head of neuroscience for a global pharmaceutical company called MSD, or Merck as it's known in the US. So the approach that we have taken, and I'm pretty sure that this is fairly standard across the pharmaceutical industry, although I think Merck was a little bit later coming to the table, is one of taking 3 steps: learning, evaluating, and executing. Now, I know that there has been an extensive program of investigation looking at every possible internal and collaborating with people externally to look at external opportunities around strategies for making some kind of therapeutic difference in the COVID-19 crisis. Of course, we are learning as we proceed, and we have engaged in a very extensive program whereby we have screened over 1,000 different compounds, collaborating, as I mentioned, externally in a whole variety of preclinical and, where possible, clinical models or environments. But I think it's also important looking forward to the future. We need to have these processes operationalized and set in place so that we're not left, as we have been in this instance, almost with our pants down. You know, this came upon us so suddenly, it could come upon us yet again. So we need our strategies to be in place so that if, or rather when this happens again, the response is much more rapid. And that's where the learning makes such a critical difference.
Thank you. Susan, do you have anything to add to that?
Yes, no, I completely agree. I think it's important to consider what's already known on a particular, compound if it's being considered for repurposing. So looking at not only the mechanism of action, reviewing the preclinical work that's been done, and looking at the important aspects that may differ, looking at a new indication. So you need to consider whether a different dose might be needed. So there are certain aspects of the early work you might need to do before proceeding. So it's clearly going to be quicker with a repurposed molecule to get into the clinic, perhaps, than if you were starting from scratch. But you can't neglect reviewing what's already been done and looking for gaps in the specific, the new indication of treating coronavirus.
So,
sorry.
Carry on, Gina.
I was just going to comment. I mean, I think, again, the approach has largely been, firstly, a double approach, a dualistic approach, in that, we've obviously looked very explicitly at whether we can target things like viral replication and activity, but also we've looked at the inflammatory processes that underlie the response to the disease. And so that's the two big, large groups, if you like, of compounds, drugs already in the clinic, already in use or in development that we've used. And then stepping further forward, we've looked at blocking agents which might block viral binding or entry into the cells. We've looked at second group, where we look at specific enzymatic functions for viral protein assembly or nucleic acid synthesis. And thirdly, we've looked at those that inhibit or block or modify the deregulated inflammatory response that contribute to the massive cytokine cascade or inflammatory response that really causes the significant morbidity and mortality. So you can look at taking this in quite a structured way, in the hope that we have things easily accessible where we have both efficacy and tolerability, as well as safety being foremost, already mapped out in order to work as quickly as possible.
Thank you. A number of the antiviral targets seem to have been inhibited by agents that have in the past been used to treat a range of other viral infections, not least HIV virus. Wilfred, could you comment on how this effort in repurposing could have impacted on the ability of HIV infected subjects to continue to access their usual treatment, and the impact of that in a pandemic setting?
Thank you. So what tends to happen is that there is a perception out in a community with regards to medicines and how, as scientists and as researchers and drug companies approach, because to someone in the community, they're going to ask themselves, okay, so why did the scientists, why did the researchers choose this particular drug to repurpose? And the perception that needs to then be managed is that people then think, okay, so at some level it must work, otherwise why is it a candidate? And so what tends to happen is that people can get ahead of the science. They get ahead of the research to then say, a drug which is already available widely within the system, once it is identified as a potential candidate or it is being used in a trial, then people go ahead and get it for themselves. Particularly in this case of COVID-19, where there are no alternatives. There's no treatment. Well, I guess with the new results that have just come out, there's hope that the dexamethasone will be a potential treatment. But in the absence of anything definitive, people in the community who are affected will look at an identified candidate as a potential alternative to nothing. And what that then happens is, particularly in our situations where the supply chains were already weak, they've been disrupted by, firstly, the epidemic itself. But what we've then seen is this massive rise in the demand for a particular drug. And as a result, the people who then require the antiretroviral, say for HIV, aren't able to access the medicines, which creates another problem in of itself, in that now you've got almost two supply chain issues happening simultaneously. One which is purely artificial, in that people perceive this to work, whereas the other one is purely as a result of just increased demand, which then results in the patients not accessing their medicines. So I think that is an aspect that as people look at candidates to repurpose and put into clinical trials, they mustn't forget that there's that unintended consequence of that particular drug then no longer being available for use in the system for routine care.
Thank you very much for those insights, Wilfred. Carol, is there particular ways in which the regulatory and GCP processes need to be addressed in the context of bringing products forward in this type of environment? And what work has been done to recommend amendments to processes that facilitate clinical development?
Thank you, Jenny. Yeah. So what we could observe in the current pandemic situation is that I believe that companies and our regulators started to learn quite fast. So previously we can observe that they had some issues with learning new things. I mean, with making a better approach to the industry and to the medicines development. But what is one of positive aspects of the current pandemic situation is that many authorities, and I'm not speaking only about the MEA or FDA, but also about the local authorities, they changed the approach, at least for the COVID patients and for the current situation. And what I can see as a project manager is that many local health authorities have prepared another like fast track for COVID medicines or COVID products. And
yes.
Yeah, so this is...
And then seminar can is required from the donor.
Yeah, and so
that's perfectly fine. If this email below from Rose is in connection with the research project, then I have not received was. What are you trying to say there?
Hello.
Then I have not received what.
Yeah.
Hello, can you go on mute, please, so that we can hear what Carol is saying? Thank you. Carol, carry on.
Yeah, thanks. Right. So the first of all, I mean, the first change we observed, and which is very positive, is this fast track for COVID treatments. And the second thing is, as you mentioned, a lot of important and very helpful guidances that have been issued by those authorities. And I know that for many companies, because almost I think all the companies had to implement some solutions to not only to the COVID studies, but also to the other trials that were conducted already or just had to start. So, and I know that the companies based their own guidances on this very well prepared guidelines. So, and I think I could also give an example, because the same was here for Poland, where 3 major associations have prepared something, frankly speaking, especially for the Polish market, but I believe which can be shared and implemented to other markets, to other countries and companies, which is specific GCP guidelines during the pandemic periods. So and such guidances, what is really good, are covering all the aspects, all the areas that should be taken care during this very specific and difficult period.
Do we think that the ethical considerations are altered in the face of the epidemic, or do the same mandates apply across the board? Is there anything that merits adjustment in that environment? Susan?
Yes, I think that the same high standards need to apply in a pandemic as would normally, so we can't compromise on that. But clearly there's some tricky issues, I think, because as Wilfred was saying, the fact that we're repurposing drugs means that in some situations it's quite easy to get hold of the medication and use it off-label as such, rather than putting the patient in a clinical trial. And so I think that is something we can learn from the past few months, is perhaps how better to manage those kind of situations, as Wilfred referred to with HIV medicines, which is also coming up with some of the medications used in rheumatoid arthritis, and was before we took hydroxychloroquine out of the mix, particularly with the malaria drugs as well. There's many unintended consequences, but as I say, with referring to the clinical trials, I think it makes it more difficult to do the clinical trials if the physician makes their own decision that they think that it's going to work and don't want to put the patient in the clinical trial, or the patient makes that decision if it's possible to get the medication off license. So I think it's even more important to keep to the ethical guidelines.
Thank you very much, Sue. Do we think that the risk-benefit balance of some of these drugs is altered if it's intended for a use for a new novel disease with a relatively high mortality rate? Or, for example, well, let's take the case of hydroxychloroquine and chloroquine treatments for malaria, widely used in the world, but with a well-known cardiovascular toxicity profile. Our experience there, I suppose, has been somewhat mixed, with widespread use on the recommendation of the US president and a negative clinical trial, or maybe even two, if you consider also the Brazilian experience. And other agents, including Kaletra, for example, which has a certain toxicity profile, particularly for liver dysfunction. Does the benefit-risk balance, is it altered under these circumstances? Feel free to join a discussion on the topic, ladies and gents.
Shall I start, Penny? So, I think that's one of the really important points, is that when you're looking at a different indication, the risk benefit that's, the risks that are acceptable in the situation of COVID-19 may be different to in the indication the medication was originally, being used for. And I was thinking particularly about the HIV medicines, actually. You're going to be taking those long term, whereas in COVID-19 it's short term. So that's one thing particularly to bear in mind. I think also the timing is very important when you're going to intervene in the COVID-19 infection, because your risk benefit, looking at hydroxychloroquine, as you say, if you're going to give it as a sort of prevention of worsening of symptoms early in COVID-19 infection, the risks that you might accept will be very different to if you're intervening when the patients are very sick later on. So and obviously the dosing also comes into that, which is why you may have a different dose, and you certainly need to look at the dosing for the different indication. Gina, did you want to comment on that?
Well, yes, I did want to comment. I think whereas the risk-benefit ratio in this instance, with COVID-19 being acutely devastating with nothing pretty much to offer to patients who are acutely ill and potentially may die very quickly, or even have prolonged morbidity and then ultimately death or long-term disability, is a major issue to take into account. However, I would say that we should not at any cost relax our strict and vigilant approach to risk-benefit assessment. Yes, we're in the midst of an unprecedented worldwide pandemic. However, we cannot allow ourselves to be sidetracked just for the sake of getting a drug into a patient who desperately needs it. We still have to very rigorously assess the risks acutely, subacutely, and chronically, and be sure that the drugs that we administer at least are not going to be more devastating than the disease itself. You know, we are, I'm sure you agree, we are bound by that desire not to cause further harm as clinicians, as doctors, as healthcare practitioners. And I do believe that although we need to prioritize absolutely our regulatory scrutiny of the processes involved, we cannot allow ourselves to administer a drug that we don't know sufficient about at the risk of causing harm. So that's how I look at this. Yes, our regulators have to be ready to process something really quickly and evaluate evidence. Yes, we're acquiring the evidence, but we need to do this with scientific robustness and rigor, because the danger is so profound if we don't. Sorry.
Wilfred, can I invite you to talk to a patient's perspective?
So I wanted to add that what we see in epidemics and what we've seen with COVID-19 is there's the theory, the theoretical aspect. Okay, so there's a good clinical practice. Let's do no harm. But at the same time, there's the reality of I've got patients who are dying every day. What do? I don't have an option. And added to the fact that as patients and as families of patients, we add to the pressure that a physician or a system has to say, okay, do something every day. It's do something. I actually think what, as people who are involved in research, we need to work out quickly is how do we find the balance between adhering to the highest possible standard, but at the same time not to be seen to be paralyzed into not doing anything? Because ultimately there's the communities and the patients will then, keep crying for something. And in the absence of anything definitive, someone will fill in the void. And we find that it's usually misinformation or disinformation. And so I think that is the challenge now.
Yeah, it is a great challenge. And I mean, looking at HIV/AIDS and the epidemic that erupted in South Africa in the 1980s and 1990s when I was actively practicing is a case in point. So much misinformation, so much need to reach out and provide something. But at the same time, we also have to be very careful that the things that we provide or the options that we provide are not problematic, are not worse than the cure or the treatment. I know that is always,
let's not forget him. He's over there on the deck.
I love the way you got it's very difficult
in there. Can I put you on the shoe balance?
Well, whoever's got a child with them, go on mute, please. But we all sympathise very much with that. Gina, you were saying.
Sorry, I know it's very difficult to strike that balance, and I'm not implying that the medical community is paralyzed, because there is frenetic and continuous activity. I've never seen things move so quickly, but the point is we cannot overstep the mark. We still have to do things that are safe and scientifically robust, because we really don't want to injure, hurt, or even kill more people than we protect. And I know that that's very difficult for the lay public often, or people who are not well versed in scientific or clinical research to understand. I've had many conversations with friends and family members about the ethical dilemmas involved. You know, someone said to me, well, why can't we all just volunteer to take this Oxford vaccine? And I said, well, you know, we can't just do that. Why can't we? And I said, well, because of the ethical dilemmas, the issues, you know, the fact that I as a physician can't condone giving someone a therapy that I think may actually be worse for them than not receiving the therapy, or even the life-threatening illness from which they're suffering. So it is a very challenging area. I'm not saying that it's easy at all.
No,
Not
very
at all.
helpful.
I agree.
That's why we're here today.
Wilfred,
Yeah.
in the context of resource poorer settings, do you think there's been equitable investigation of repurposed medicines in that setting, as opposed to the richer countries of the world, or what more could we or should we be doing?
I think we've seen an effort definitely to include resource limited settings in some of the studies. I think I should mention here that as researchers and people who are involved in studies, we've been set up to patients and to the public in that there's a narrative that has gone out there that in an epidemic, look to the scientists, and the scientists will very quickly work out what's wrong and they'll have a solution. I mean, if you look at all the movies and all the stories that are out there, that's how we've been framed. And as scientists, we haven't really rebuffed that. We haven't gone to the movies and said, guys, it just doesn't work like that. And so there's a public perception that, you know, the people on this call are smart enough and have enough resources to figure out an answer to COVID. And it's particularly dominant, I find, in areas where information doesn't really flow that well, which is why I think your question is important, to make sure that we leave no one behind. No area is left behind, whereas be it resource rich or resource limited. And I think the other aspect that we shouldn't forget as we do this is this issue of unintended consequence, because to speak to what Gina was saying, we have people who will say they frame it as a dichotomy. Do I die now from the pandemic, or do I die later or from an experimental treatment, which may or may not help me? Which is better? And so I think every time that research is done in any area that it's done, as scientists, as researchers, we actually have to go out and continuously engage the community so that we make them understand that this is how it actually works. This is how clinical trials actually work. This is how research is done. It takes a process, and there are guidelines and there are principles and there are things to be done. And so that the community not only buys into what is happening, but they fully understand, and finally, they also feel a sense of ownership with the whole process.
Yeah, I think that's really important, and not just in the context of dealing with COVID-19, but as we mentioned earlier, all the other intercurrent illnesses, clinical trials and research that are ongoing. And you mentioned it, and we have been very much keen to enforce the strategy of no patient should be left behind. So we have been implementing huge outreach programs to ensure that patients involved in other clinical trials, other programs, whether that is HIV, infectious disease, neuroscience, oncology, particularly where people again may face life-threatening illness, they are able to continue with their clinical trials and resource. We've been able to initiate huge strategies to take drugs to patients in their homes, to monitor and facilitate administration and monitoring of response or side effects in their homes. This is unprecedented, especially in the oncology space, and yet somehow we have been very successful, I might add, at least in terms of the Merck MSD experience, in actually allowing clinical research to carry on so that no patient is left behind. It's been a monumental effort, and I think many patients are starting to understand that process. Yet again, as Wilfred rightly says, though, there is a huge need to educate people about why this is the case and how it's being done, so that they are empowered as patients, but also as members of a community, to be involved and to have a voice.
Thank you. I'd like to turn now to sort of novel medicines development for a specific condition in this context. So again, for the panel to think, what are the key elements of drug discovery and the preclinical investigation that we normally conduct for completely novel discovery of medicines that can help rapid development of medications for this condition? Who would like to start?
I can start. Maybe I'm talking too much. Okay, well, there is a lot that is being done, and clearly the main approach is obviously to look at platforms that already are in place, because no one wants to reinvent the wheel. It takes too long and the risks are significant. So in terms of looking at the vaccine space, there are a multitude of different approaches, and people are obviously looking at things that have worked in the past. For example, using strategies like the recently developed Ebola virus and that platform, which allowed for a relatively short 5-year process to take place from the time of inception to approval, which is unprecedented. So can we track onto established processes and use them? Or do we look at different approaches in that space? Or additionally, do we look at other agents, not a non-vaccine approach? And therefore, do we look at things that can be administered orally rather than intravenously, that can be singly dosed rather than multiply dosed, that don't require a cold chain for transmission? Again, looking at many parts of the world where cold chain transmission and supply is an issue. And then, of course, we look at the manufacturing process, the upscaling and development. So all these things are factors that are being taken into strong consideration as we start from almost step one, where we're looking at developing totally new vaccine or non-vaccine drug-based therapies, whether these are antibodies, small molecules, etc.
And has anybody done very much in the computer simulation type area to look for binding sites on various proteins? What have we been able to leverage prior understanding of different viral diseases to think about logical strategies for antivirals? In the inflammatory space, we've generated quite a lot of information in different areas. I mean, if in oncology, for example, the cytokine storm that follows cell therapy has been an interesting paradigm, I guess, not linked to the inflammatory response in this condition. Did we start from a higher base, do you think, than we would have done if this had just erupted very suddenly with no prior practice, as it were? Sorry, Gina, we can't hear you.
I mean, I do believe we have established precedents. I don't think we are as ready as we should have been. And sorry, can you hear?
Yes.
No?
Yes.
Can you hear me? I have unmuted. Oh, I was saying that we are using established technologies, things that we have already generated in-house, and there is a huge amount of collaboration between academia, industry, NIH, the FDA, EMA, companies, FPM in Europe, to try and really make a difference and use established or known therapeutic or analytical approaches. But I do believe that we could have been more prepared. We could have been further down the line in the event that this kind of thing happened. We could have been more prepared, and hopefully moving forward we will be. It's just sad that this had to be our learning point. But I mean, what can we do?
Yeah. So I just want to echo that, to say part of it is learning from prior experiences and finding ways to be able to activate where necessary all these networks, these collaborations, making use of what's already existing, mobilizing the resources, and then being able to scale up very, very quickly. I think I don't know, it may be debatable, but in my mind, the possibilities of another pandemic like the one we're going through happening in the near future is very real. We've learned, from the first SARS outbreak to Ebola, now to COVID-19, important things have been learned. And I think going forward, I agree with Gina that there shouldn't be any excuse for us to be unprepared. The real question I have would be, can we dedicate the time and the resources to set up the foundation that in future we're able to get on top of the situation and scale up very, very quickly?
I suppose when you think about the colossal economic impact of this disease, and in particular the public health measures that were taken with shutting down of significant areas of commerce and significant job losses and all that has entailed, that there's an argument that a relatively lower amount of earlier investment might have put us in a better position. In terms
yes, I would. I think so. I think I agree. It would have, and certain national or even global programs in the last years or even decades have been down-regulated or down-prioritized because we thought it would never happen to us.
Mm.
We let our guard down, I think. I mean, we as a global community, which is a sad thing. I mean, I just look at the response of many African countries who are more used to dealing with epidemics like Ebola, for example, in terms of the strategies that they have been using to track and trace people coming into the country or even moving around the country. They've, in some respects, in many respects, been much more prepared and much more effective than many European or US or North or South American based countries, or even maybe in some places in Asia are used to this and others are not, and you can see a difference. Yeah, I don't want to get sidetracked, but
Susan.
Yes, what I was going to say is I think in order to be more prepared next time, I think we can probably learn, globally it can be learned from the UK investigators, the recovery trial, because my understanding is they already had a protocol virtually ready, set to go when the pandemic started, which is why they're now getting answers in advance of studies that have been done in other regions. So I think, and also I think the other lesson we can learn is the amount of collaboration internationally and also between industry, academics and clinicians that's happened. I think we can hopefully now there's the understanding that it is important to be prepared in advance, and there can be protocols set up ready to go very quickly to get these answers coming through about which therapeutics work, and also have some idea of what could be tested in a similar situation in the future, the antivirals and the anti-inflammatories we've talked about.
Do we think
thanks. I'm not sure if you will agree with me, but looking at the past 20, I think, years, global population have faced a lot of different pandemic, which was just spread within a region. So frankly speaking, a lot of the companies, and at least the smaller ones, should have some solutions hidden somewhere. And as you mentioned, due to economic reasons, this has been delayed or at least stopped for some time. And in my opinion, the only difference currently is because we as a population, as a global population, we're prepared for such situations. And the only change, the only difference was a global impact. global impact and lockdown, which face also other areas of our life, and this is only difference. And frankly speaking, looking at the past history, and not only the past 20 years, but far later, Ilya, so a lot of different similar situations has came from Asia mostly. So frankly speaking, the future drug development, the same as it is going back for the antibiotics, the same, we need to think about new antiviral drugs. And I think it is a future, and this is not a question if it's happened, but if it will happen and how strong it will be. Yeah, so I think this is the best, having the current development of many areas. This is, I think, the best situation and the best moment to learn from that, and to prepare for another thing which I believe could face the world within even next 10, 15, 20 years, which is thus the fact.
So we touched on differences between countries and ability to transport medicines in a relatively simple way. Does this include the route of administration of a medicine? Are there cultural differences in the acceptability of different types of routes for administering a product? And how do we cope with phase 1 trials in this kind of situation? In an oncology setting, of course, we do our phase 1 trials in the patient population. Is that the same here, or do we have to go through some other routes to speed things up?
Yeah, so the issue of route of administration, I think, is key, because you want to look at supply chain systems and health systems which are already suffering from an inability to get product to a certain place. Let me give an example. So let's say you have an injectable product which needs to be stored which needs to be transported at cold chain and stored in a fridge. You then want to take it to a place where the transportation facilities are missing. The system itself doesn't have the facilities in which to store the product. And you could even have something as simple as there's just no power. So this then makes it difficult to take such a product to the people who need it most. But at the same time, I understand that if we focus too much on the route of administration, rather than does it work at the beginning, then you may actually be stuck to say, we only have something that works, but it's only given intravenously. Does that mean we don't proceed with doing the study? And my answer would be no. We need to ensure that the drug works, it is safe, and we've done the studies in a way that is ethical. And then I think as we go forward, we can then start to focus more on how can we change the formulation to make it easier to administer, easier to transport, and easier to store.
There are several antibody therapies which are starting out in clinical trials that have been designed based on the sequencing of type-specific antibody from recovering patients. Do we think that the rapid startup of these clinical developments of these has been facilitated by the work done on antibody therapies in the recent decade? In other words, have we been able to leverage our understanding of the production of these and their administration efficiently because of that experience?
Yes, I think so. I do believe that we are much more easily and effectively able to move faster because of the learnings that we have. We know what we should and we shouldn't be doing. We know what signals to be looking for. We have established assays that are ready to go, if you like. But of course, at the same time, there are things that have to be set up de novo, because this is a unique pathogen and a unique approach. And clearly in antiviral space, it's an area that we've had many success, but we've also not always got on top of things. Not wanting to sound unduly pessimistic, this is not an easy area. I don't think any area is, to be honest. So there are some viruses that we can treat or we can develop vaccines for, but there are others that we can't. And hopefully this is not one. I've heard a lot of optimism that there's a lot of reason why we think we can develop therapies or vaccine-based approaches against COVID-19, or rather SARS-CoV-2. But, as with all clinical research, the proof lies in the clinical data. You can have the best preclinical assays, models, development program in the world. Your phase 1, even your phase 2 can look pristine and fantastic, but it's really when you get into many patients in the real world, that's where you see the value or not of what you have done. And unfortunately, I don't think we'll be able to get away from that. I think we'll get there, and we'll get there really quickly. Will it work? Will they work? I don't know. But at the moment, we're also making many attacks on the virus from many different ways, taking many different approaches. So I'm hopeful that at least one of those hundreds, if not thousands, will do something significant.
Thank you.
Yeah, just to add to that, I think we've got lessons learned from administration of monoclonal antibodies in HIV, particularly for prevention. I'd just like to add that one of the lessons that arises from administration of monoclonal antibodies in the space of HIV has to do with consent. So, and primarily the issue that researchers are aware of, which is therapeutic misconception. And I think it's things we've got to work through so that we're sure that it's not other confounders which are affecting the efficacy of the product. Because you may find that depending on how the communities understand the whole concept of monoclonal antibodies or therapies, antiviral therapies that we're developing, they may either refuse to sign on but withdraw, sign on and not participate in a way which results in anything meaningful being drawn out. Again, it's an area that as researchers we kind of avoid, because we just want to focus strictly on the science, not knowing that the science does defend. Yes, it depends on the patients and what the patients do. If they don't come, if they do, if they refuse an injection and they want a tablet and you've got injections only, then again, your science can't move forward.
In the last few minutes, sorry, I was wondering if it's possible to address some of the questions coming from the attendees. Thanks.
I can't actually see questions from the attendees. Read them. Okay. Right. So let's have a look at William Payne. Let's find William's question. He's asking, is the risk benefit always clear cut, and does expediency sometimes permit the taking of risk, which in the best conditions would be regarded as unethical?
I don't believe that the risks are always self-evident. I mean, we have lots of examples in drug development where, I'll give you a case in multiple sclerosis, for example, monoclonal antibody, daplusumab, a new approach in multiple sclerosis, looked very good in phase 1, 2, and 3. We took it, it was approved, and then we saw a handful of sporadic meningoencephalitides which were fatal in a handful of people. But of course, that is not a risk that is tenable. So both the EMA and the FDA withdrew the drug quite correctly, but we saw no evidence of those issues in the clinical development program where thousands of patients were subjected to the drug. So, you know, that's just a case in point, but it can happen, and it can happen anytime, anywhere. So, you know, we can only do our best to identify the risks upfront as we move forward within the context of our clinical program, and hope that we do more good than harm. That's what I say anyway.
Yeah, and I agree. This is Carol. Sorry. Yeah. So, and I agree with Gina, because we cannot just apply clinical setting, clinical studies into the real world. I mean, because we must remember that for clinical trials, we are trying to enroll, we always think, almost healthy subjects. So, you know, without other disease, just with the disease we are looking at medicine for. And that's the very important thing here. So we all know that in such situations like we have now, this risk-benefit balance is affected. And I think, in my opinion, we should go two ways. So first of all, of course, use clinical trial setting for searching for new and repurposing medications. And from the other part, we need to, I mean, the doctors, the healthcare professionals need to use currently available documents and data which is shared globally for their decisions. So we need to just separate the healthcare professional decisions and the clinical trial setting.
Marcia,
so
just to add to that question?
Yes, I wanted to add to that.
Oh, sorry. I was going to say, because the time is running out, Yolanda Alvarez, her question was, there have been several strategies. What do you think about the herd immunity proposed by the government at the beginning, but never was put in place?
Well, I don't believe that they could have put in place a herd immunity. I mean, herd immunity implies that a significant percentage of the population is exposed and is infected. You know, measles, we require almost 90%. This virus, we think maybe 60 to 70%. How can you apply herd immunity to a naive population? Ultimately, I think, years down the line, yes, there will be baseline exposure in the population, but that will not come for a long, long time. The exposure has to be significant, the fatality has to be significant. And I would not be prepared to take that risk up front of literally sacrificing many people up front in order to achieve something like that, which will take years.
Wilfred.
So I think the issue of herd immunity for me is an important example of communication, and communication around a particular idea or principle that some scientists have. Because when the issue was then put to the public, it appeared as if there was consensus, or at least a majority of the scientific community was behind the issue of herd immunity. But this wasn't the case. And I think as scientists, we also need to be able to say, even though we put forward a particular course of action. So for example, if we say we're going with herd immunity, we must communicate it in such a way that this was one of many things that was put on the table. And this was chosen for the following reasons. It is not the only thing that was discussed, and it is not the only thing that was, or that has the majority of scientists behind it.
I guess the question is whether it merits a debate with the public in the context of empowered and democratic institutions. If that was going to be seriously pursued, that was an area possibly that required public consent in a way which, you know, that in a more dictatorial society or wouldn't be thought about. And to me, this came across as being, if this had been influenza, it might have been a good idea, or something like epidemic influenza, it might have been a good idea. But in the context of a disease with a 20% hospital admission rate and a 50% in-hospital mortality rate, as it appeared at the early outset, the idea that the public was going to agree that allowing them to get the disease willy-nilly, like we did with children when I was a child, chickenpox parties were all the rage, for example. And now we have a vaccine. Yippee. My grandchildren don't have to have chickenpox.
We have the concept of immunity in the absence of a vaccine is a problem.
We have 2 more questions, and we're over time, so it's up to you if you want to respond to them before we close.
Yes, let's finish with the 2 questions and then we can close.
So one is from our president, Professor Tim Higginbottom. What place does an understanding of the pathological changes in the disease play in determining therapeutic targets?
Can I answer, or does someone else want to?
Yeah, carry on, Gina.
I think an essential place. You know, we recognize this very early. I mean, we were getting reports in January from Wuhan about the fact that it wasn't the virus that was killing people; it was the response to the virus. And if we hope to achieve a therapeutic response that makes sense, then we really need to understand how the virus is acting, you know, how the immune receptors, the AC2 pathway, the cytokine storm generation, the microthrombosis as part of an inflammatory cascade works, the impact on multiple organs resulting in multiple organ failure, not just on the lungs, the impact of cardiovascular risk factors, morbidity, etc., on a virus that seems to be very much endothelial receptor tropic, therefore having an effect on the vasculature, and for where I come from, the neurovasculature, so leading to stroke and all kinds of problems. You know, it's not just treating the virus, it's treating the response and also be able to intervene where someone has a stroke, myocarditis, renal failure, etc. You know, microvascular thrombosis, can we get on top of that almost immediately? Because then we can almost preempt that in the patients that are in the hospitals and try and prevent these complications from ensuing in the absence of something that wipes out the virus or ups our immune response so we don't get it. Okay.
I think that's a very comprehensive answer. Marcia, last question.
Last question from Faheen. What is the status of remdesivir in this situation? There was some news regarding its approval within NHS for severe cases and ongoing price negotiations.
Who knows about remdesivir? I don't know personally. Susan.
So I'm not sure I can answer on the pricing bit, but I think the status is that there was evidence from one of the trials, I think it was the Gilead trial, that it reduces the time to recovery. And I think the MHRA have now issued, I think it's called EAMS, I think it's expanded access, so that it can be prescribed. And in fact, I know that the clinical trials ongoing are now allowing in many situations that it's part of standard of care. But I can't really comment on pricing. I don't know whether anybody else on the panel can comment on where it is with that.
No, Gilead have given some indication that they don't intend to charge a very large amount of money for this, but they're struggling at the moment with manufacturing to meet demand, I think. The early access to medicine scheme required that Gilead actually apply for that access, and they were somewhat surprised by the amount of demand that there was after that first grant by the MHRA. Of course, in the US, the MHRA followed on from the emergency use authorization in the US, which is not a proper authorization of the medicine to be used in widespread clinical practice, but as defined within the protocol, which is a a package insert type information set, which is available to clinicians. And in the US, similarly, there have been some arguments about supply to the various states and competition, which is one of the more distressing elements. It is an intravenous medication, so it can only be given in a hospital setting. We're not set up in this country for administering IV medicines at home as much as in some other parts of the world. And maybe that's something we'll have to consider for the future, because an antiviral is best used as fast after first recognition of illness than late. And indeed, the outcomes in the remdesivir trials have all indicated the earlier it's used, the more likely it is to truncate disease and prevent later complications. Excellent. Well, I think thank you to our audience for raising those questions, and thank you to our panelists who have made for a very interesting discussion this afternoon. We have the third in our series of webinars on the 1st of July. And please, if you've enjoyed this one, please sign up for that one, when we will be dealing with clinical research and development for vaccine medicines. And registrations are now open. I'd also like to thank all our panelists for their active participation in the discussion today, and thank you very much indeed, in particular to Wilfred, who's been sitting up late in Zimbabwe, to Susan, to Gina, and to Carol, who we couldn't see, but who did actively participate. I'd also like to thank the organisers of this meeting, the folks at the faculty who set this up and managed it for us. And I'll bid you all good day now, and hope to see you again at our next webinar on the 1st of July.